the compounds that have at least 20% inhibition, and we screen compounds in quadruplicates, and we use at least 3 standard deviation difference between compounds in our vehicle control. And we discovered 22 anti-infective compounds and 44 pro-infective compounds. So here you can see the growth curves for our vehicle control. This is the fluorescence, and it's observed over time for 72 hours. It's a mycobacteria growth. And you can see that when we add our compounds, we observe inhibition of the growth. And this is our heat-like activities. This is our positive control with formaldehyde at 20 micromolar concentration. So here you can see the results of our screen with the compounds sorted by the pathway. And you can see that we observe some of the anti-infectives activities. At the same time, we observe some pro-infective activities, which are also quite interesting. This is the research in this direction could be interesting because it can show us the pathway crucial for the defense against mycobacteria. So we can see which pathways are inactivated when we expose our infection to the proactive compounds. So here we have pro-infective compounds. So here we have a distribution of the hits that we get. You may notice that we have quite a lot of the hits from autophagy pathways, and there is some tryptophan pathways, and it's cation channels. And we have these enrichments both in pro-infectives and anti-infectives, and this just display the crucial importance of autophagy in host-pathogen interactions. For example, the xenophagy, which is clearance of infection with the use of autophagy. So here there are some examples of the structures that we get. So you see the percentage of, I forgot to mention that although we think that these compounds belong to this pathway, it's not necessarily true because it's only putative interactions, and that means that the compounds actually may have completely different activity. So it's just assumption that we have from the in silico studies. So now I presented you the primary screen results. So now I will switch to the validation steps where we made the validation in Academia Castellan and Dictyostelium infection assays, also in antibiotic assay with Mycobacterium marinum growth inhibition, and in Dictyostelium growth inhibition assay. I present you the results. So here we observe about 18 anti-infective compounds, and you can see that actually we have a strong enrichment of anti-infective activity in our assay. At the same time, we observe some antibiotic activity which doesn't display much activity in vivo. This is one of the problems of traditional screenings in broth. And of course, we are interested mostly in this part of the graph of the diagram because it's we don't want the compounds that have strong cytotoxic activities or antibiotic activities which are not working in the host, inside the host. So after our amoeba screening, we need a validation step. We need to validate our system in the mammalian system because we don't want to discover the drugs that are purely amoeba specific. We want to generalize our data for the other organisms, particularly for the macrophages. So for that we use the cultivated macrophages of microglia cells, PD2 cells, which are basically a brain macrophage. And in order to monitor the activity in these macrophages, we use high contact microscopy. So you can see it here. There is a scheme of the 96 well plate. We monitor the changes of fluorescence in each well. So the green represents the bacteria. You also can see some of the cells here. So I will show you activities that we observe. So here the example of the control, basically the vehicle control. You see the infection, and you see that the amount of bacteria are not decreasing over time, it's increasing over time. We also observe some of this cytotoxic activities on the cells. But if we look at the antitubercular compounds, like rifabutin, we observe no increase of the fluorescence. That means that there is no increase of the bacteria numbers. At the same time, we see that the cells are feeling fine. We don't observe much of the cytotoxic activities, and we are really interested in the compounds that display such kind of activities. This is the movie that was shot during the 72 hours. So here there is a scheme of the plate, 96 well plate, where each of the square represents one microscopic picture from these wells. And you see the controls here, so the vehicle controls, and you can see that we observe various type of activities in our screen. So we have control-like activities, so basically not much effect of the compound. We have the anti-infective activities and some pro-infective activities where we get increase of the bacteria numbers. And we observe also cytotoxic activities. That's how it looks like with the magnification. So this is the control, this is the our anti-infectives activities, this is the pro-infective activities, and this is the cytotoxic activities. And here you can see that if you just look at the fluorescence, you may think that this is a hit because here we don't see the increase of the fluorescence comparing to the control. But at the same time, we see that the cells are not feeling very well. It means that there is a strong cytotoxic activity, but it doesn't mean that we cannot use this kind of compounds. There is still a possibility of therapeutic window. So when we have a strong cytotoxic activity on the bacteria and less strong activity on the host. So that's why this kind of compound are still need to be analyzed. Also, you can see the quite peculiar pro-infectious activities in some of the cases. So this is our anti-infective activities. Another movie from anti-infective activities. Again, it's 72 hours time-lapse microscopy, and you can see that we observe lots of intracellular bacteria and almost no extracellular bacteria. The cells are feeling fine, and there is no increase of the mycobacterial load in the cells. So here, right now, I will present our data from other libraries. So the GSK TB set. So this is an illustration of the data that we are getting from the GSK screen. You see that we observe, this is the vehicle control. So results normalized. So everything is normalized to the vehicle control. So vehicle control is 1. We have anti-infective compounds, pro-infective compounds, and also our control. And you can see that many of the compounds display anti-infective activities, although many of them are not very strong anti-infectives. So this is a result from malaria books library. You can see here you have relatively, this is antibiotic assay, this is the anti-infective assay, and you can see that you don't observe much of the anti-infectives. This is the every plate sorted, and we have basically only one strong anti-infective. So here the summarized result of the 3 libraries. You can see that we have many anti-infectives in our Synergia library, which confirms our ideas that it's host pathogen oriented. At the same time, not surprisingly, we're not getting much of the hits in the malaria box because it's oriented on another pathogen. And in GSK TB set, we get lots of antibiotic activities, which also can be expected. But surprisingly, we don't get much anti-infectives, so there is not much strong anti-infective compounds. So many of the compounds display very low inhibition rates. It's like 10% or 20% inhibition. One may think that the problem is with the host, so maybe it's just Acanthamoeba specific, but actually we observe similar activities in our BB2 assay, so our macrophage-like assay. And you see that although we still get more stringent results for this assay, the overall pattern is similar. So apparently many of the antibiotics doesn't display a strong anti-infective activity. So after finding our hits and validating them in macrophage-like assay, we performed rounds of studies, structural activity relationship studies. So we established a set of chemical derivatives with the help of Professor Scaposa research group. So this is some of the results of these studies. This is a chemical family from GSK library. You can see that these compounds have similar chemical scaffold, but depending on the changes in the chemical groups, we get different activities. So that using this data, we can estimate which chemical groups are the most important for the activity of the compounds. So here there is some of the examples of the SAR data that we are getting, and with the chemical compounds, we will not talk much about it. So now there is a conclusions. So we developed our assay using amoeba and Mycobacterium marinum as our host pathogen model system. We established the assay to validate our hits in dictyostelium and mammalian microglial cells. We performed a medium throughput screening. We identified about 22 anti-infectives in Synergia library, 18 infectives in GSK TB set library, 1 anti-infective in malaria box library, and we validated our assay in dictyostelium mammalian microglial cells, and we performed some preliminary SAR studies. So what is the possible perspectives of the work? It's a host pathogens project which will continue to be in our lab. So there is this project is based on the dual RNA sequencing of the both host and the pathogen using dictyostelium discainum and Mycobacterium marinum with analysis of transcriptional profiles in the presence and absence of antitubercular compounds. That follows by metabolic and signaling and regulatory pathway analysis and modeling, and mechanism of action and identification studies. With that, I would like to finish and to say big thanks to my lab and the research groups that we are collaborating with, all members of Synergia project, and especially Sebastian Kika, a postdoc in our lab with whom we was working in this project. I'd like to thank NCCR platform for great help in high content microscopy. And I would like to say some additional thanks to the members of our lab. It was really great pleasure to be in this lab. It's a very great atmosphere. People are always helpful, and it's really fun. I would like to say thanks to everybody in our international PhD program, and I would like to say thanks to my friends and family for always supporting me, and I would like to say thanks to all of you for listening to me. Thank you for your attention.
So, the queue, we now go now with the questions. We'll first ask Professor from the University of Munich, of course, the Ludwig University in Munich, to start with the questions.
Well, thank you very much. It was an impressive amount of work that went into this screening now. In a way, the fun part will only start now. So if you would have to do another PhD thesis in theories now, what would you pick and how would you proceed? The last point was identify the mechanism of one of these hits you have in your many screens. How would you proceed and what?
So I would gladly continue the mechanism of action studies. For this, the already mentioned part4 project is a nice alternative. Also, I think that the pro-infectious compounds are also very interesting subject for the research, because this is some of the activities that are usually not being addressed much and considered as a negative data. So it would be really interesting to analyze both the anti-infectors and pro-infectors. And I think that a transcriptomic approach is a good start for the mechanism of action studies. At least it will help to identify the target pathways. And after that, I think we can narrow down to the more precise molecular targets. For example, with the use of mutants targeting crucial pathways, signaling pathways, and metabolic pathway in the cells. For example, mutants of autophagy could be interesting to use something like ATG1 epithelium mutants to see whether the autophagy affects the have an impact on the activity of the compound. So, combining the exposure of the cells